In this video, we're going to look at the hormonal responses to anaerobic training. We'll explore the responses of key hormones like testosterone, growth hormone, IGF-1, and cortisol during various anaerobic exercises, such as sprinting and cycling, whilst looking at repeat sprint and high-intensity sprint efforts. Additionally, we'll look at training studies that discuss the factors that influence these hormonal responses, such as training volume, intensity, and rest intervals. Steroid hormones play an important role in regulating neuromuscular performance and have been found to modulate neuronal activity in the central nervous system. In vitro studies have demonstrated that testosterone and cortisol can induce rapid changes in neuronal inhibition or excitation within different regions of the brain. Although hormone-induced changes in behavior and cognitive function are more delayed in humans, further research is needed to fully understand the underlying mechanism. Athletic studies in sports such as soccer, sprinting, handball, and rugby have shown that testosterone and cortisol concentrations correlate with running speed, jumping power, and strength. These findings suggest a potential influence of testosterone on the development of type 2 muscle fibers. The exact mechanisms responsible for the observed differences in neuromuscular performance are still being investigated, but it's likely that testosterone and cortisol affect both the central and peripheral neural networks involved in human movement control. One form of anaerobic exercise that is regularly performed in the training of athletes is high-intensity interval exercise, which is a time-efficient alternative to combined endurance and high-intensity exercise. High-intensity interval exercise involves short-duration bouts of exercise performed at high intensity, typically above 85% of VO2 max, followed by specific recovery intervals. High-intensity interval exercise can be classified into two distinct exercise formats: high-intensity interval exercise and sprint interval exercise. High-intensity interval exercise is performed at intensities between 85 and 100% of VO2 max, with bout durations ranging from 12 seconds to 4 minutes. A sub-classification of high-intensity interval exercise is sprint interval exercise, which is reflected by an all-out effort, corresponding to intensities exceeding 150 to 200% of VO2 max, with bout duration ranging from 5 to 30 seconds. From an energy systems viewpoint, during the initial bouts of high-intensity interval exercise, phosphocreatine degradation and glycolysis are the primary sources of energy supply, with glycolysis contributing to over 80% of the energy. However, as the exercise progresses, oxygen-dependent metabolism plays a larger role in energy production. Both methods have been shown to effectively activate oxygen-dependent metabolism and are considered effective methods for improving physical performance and health. As this video is all about understanding the hormonal responses, we will now focus on a few studies that highlight the hormonal responses to anaerobic training and high-intensity exercise. The majority of these studies focus on either running or cycle ergometry. The first study that I'm going to highlight is from Meckel et al., who examined different types of sprint interval training for their effects on the anabolic and catabolic hormones. In this study, 12 healthy elite junior handball players completed 2 different sprint interval runs on a treadmill. The first protocol consisted of increasing distances, that's 100, 200, 300, and 400 meters, and the second protocol was of a decreasing distance, going from 400 to 3 to 2 to 100 meters. Their speed was set at a constant work rate of 80% of their personal maximal speed, based upon their own maximal 100-meter sprint speed. If we look at the results, both types of sprint interval trainings led to a significant increase in lactate and the anabolic growth hormone, IGF-1, and testosterone, indicating an anabolic type hormonal response. Calculations of the area under the curve indicated that lactate and growth hormone levels were significantly greater in the decreasing distance exercise session. That's the 400 meters going down to the 100 meters. In another study by the same research group, they used a 4 by 250 meter sprint protocol with a 3 minutes rest period between efforts, whilst running at 80% of their maximum 100 meter sprint speed. This protocol resulted in significant increases in growth hormone and IGF binding protein, testosterone, and the testosterone to cortisol ratio. The protocol had no significant effects on cortisol and IGF concentrations, and therefore the authors indicated that this type of training led to an anabolic type hormonal response. Shifting our focus from running to cycling-based anaerobic exercise, a protocol of 6 by 8 second sprints interspersed with 30 seconds of recovery on a cycle ergometer resulted in significant increases in salivary testosterone and cortisol concentrations. In other cycling studies that looked at measuring growth hormone, a higher mean serum growth hormone concentration was observed after a 30-second sprint, which was 450% greater than after a 6-second sprint. When we look at the difference between the 30-second and the 6-second sprint, growth hormone also remained elevated longer after the 30-second sprint, reporting a significant elevation on average at 90 minutes post-exercise, compared to approximately 60 minutes after a 6-second sprint. There's also a large inter-individual variation in human growth hormone response after a 30-second sprint, and we have to acknowledge that as well. Although the mechanism for the difference in this growth hormone response is still unclear, the authors hypothesized that the duration of a bout of maximal sprint exercise can determine the magnitude of the human growth hormone response. Further to this last point, and this is perhaps a major contributor to large individual variations in results observed in these studies, is that there are multiple factors that need to be taken into consideration when extrapolating the results that we see, and therefore we need to take these considerations in how we program. A meta-analysis on the effect of high-intensity anaerobic exercise and growth hormone response highlighted that the mode of exercise may affect the growth hormone responses. For example, treadmill sprinting, where both arms and legs are active, has been suggested to elicit a greater growth hormone response than sprint cycling, where movement is concentrated in the lower limbs. Age also appears to be an important modulator of the growth hormone response to exercise, with young individuals exhibiting a higher growth hormone increase after sprint exercise. Frequency of training and session planning also play a role here as well. This has been highlighted by the fact that a repeated bout of sprint exercise on the same day, i.e. 2 sessions within the same day, results in an attenuation or even disappearance of the exercise-induced increase in growth hormone. Nutrition plays a role in hormonal response and performance as well. To illustrate this, a study investigated the effects of caffeinated chewing gum on fatigue and hormone response during repeated sprint performance. 9 male competitive cyclists completed 4 high-intensity experimental sessions consisting of 4 sets of 30-second sprints. That's 5 sprints each. Caffeine at 240 milligrams or a placebo was administered via chewing gum following the second set of each experimental session. Testosterone and cortisol concentrations were collected at rest and after each set of the sprints. As we may expect, caffeine ingestion led to a 5.4% performance enhancement compared to the placebo condition. With respect to the effects on hormones, salivary testosterone increased rapidly from rest, around 53% in all trials, with an additional 12% increase following the caffeine treatment. In contrast, cortisol concentrations were not elevated until after the 3rd exercise set. However, the ingestion of caffeine resulted in a 21% reduction in cortisol concentration relative to the placebo. These findings indicate that acute caffeine ingestion attenuated fatigue during high-intensity sprint exercise and was associated with elevated testosterone and decreased cortisol concentrations. In support of these findings, acute increases in testosterone are reportedly related to exercise selection and intensity, nutritional status, and experience of the athlete. Testosterone responses are also related to the mechanical stimulation of the muscle, that is, exercise protocols which involve a greater amount of active muscle mass with higher loading, that's greater time under tensional force, eliciting a greater testosterone response. It is possible in relation to the study that caffeine acted on the central nervous system to stimulate a greater recruitment of motor neurons, which resulted in a greater degree of active muscle mass. Subsequently, a greater force was produced and testosterone concentration was therefore increased. Additionally, an acute increase in testosterone can directly influence force production by facilitating neurotransmitter release. Thus, the ingestion of caffeine together with acute elevations in plasma testosterone may act synergistically on the central nervous system to directly increase force production and improve performance. In contrast to the rapid increase in testosterone, cortisol did not increase substantially in either the placebo or caffeine condition until after the third exercise set. The timing of this increase is in context with the dynamics of cortisol release via the hypothalamus pituitary adrenal axis. Interestingly, caffeine ingestion led to a blunted cortisol increase relative to the placebo treatment. This may benefit the athlete, as previous research has reported that suppressing cortisol during exercise is associated with greater gains in muscle mass. In conclusion, hormonal responses to anaerobic training and high-intensity exercise are complex and multifactorial. They are influenced by various factors including exercise intensity, duration, mode, and individual characteristics. Understanding these hormonal responses can help optimize training protocols and may also provide avenues for additional strategies, such as nutritional interventions, to enhance acute hormonal profiles that are beneficial to short-term training performance with a goal to enhance long-term physical adaptations.